As mentioned, microbes have been extremely helpful in providing us with medicines that we need. This includes insulin, depicted here. Human insulin is needed increasingly every year by more and more diabetics in the United States and worldwide. In the past, insulin needs were met by harvesting pig and cow insulin from butchers that were butchering pigs and cows for meat. However, this procedure was expensive and would come with the possibility of transmitting infectious disease from the pig and cow to the human in the process. Despite those issues, this is how insulin was manufactured for decades. But with today's diet and exercise routine, many people in the United States are at increasing risk of coming down with type 2 diabetes and needing insulin shots every day. So the needs of insulin by humans has skyrocketed. In turn, genetic engineers came up with a solution to this increased need. Genetic engineers were able to find human insulin genes from a human cell, and they took these human insulin genes and put them into a bacterial cell, E. coli. E. coli then, in huge vats around the United States, are producing human insulin for diabetics. So diabetics today are now receiving human insulin instead of pig or cow insulin. And this production can continue indefinitely. Other chemicals are also produced this way, including human growth hormone and some vaccines, including the hepatitis B vaccine. The agricultural industry also uses chemicals produced by bacteria in processes that result in what are known as bioinsecticides. Bioinsecticides are pesticides produced by living things. Probably the most famous of these is Bt toxin, made by the bacterium Bacillus thuringiensis. Bt toxin has been used by organic farmers for quite some time because it is a pesticide that was harvested from bacteria, a naturally occurring source. However, Monsanto, a giant agricultural company, was able to identify the gene that makes the Bt toxin and genetically engineer that into some plant species, including corn and soybean. These plants are now resistant to pests such as caterpillars and beetles, all a result of inserting a microbial gene into a plant. Human use of microbes did not end there. We also use them occasionally in processes known as bioremediation. Bioremediation is the act of adding materials to the environment, such as fertilizers or microorganisms themselves, to increase the rate at which natural biodegradation of contaminants occurs. Natural biodegradation of contaminants such as petroleum spills or radiation would occur on their own, but would be extremely slow. Instead, genetic engineers and scientists have found that these processes can be sped up by bioremediation procedures. This was used in the Exxon Valdez oil spill of 1989. Some beaches were cleaned up using bioremediation. In this case, fertilizers were added to rocks and soils on beaches in order to encourage the growth of microbes that would eat the spilled petroleum. This is technically referred to more specifically as bioaugmentation. Another, more massive spill occurred just about 20 years later in the Gulf of Mexico in 2010. This oil spill was about 20 times more devastating than the Exxon Valdez incident in 1989. The spill was the result of oil gushing out of a damaged drilling rig called the Deepwater Horizon. This damage ended up killing 11 people on the oil rig and injuring 16 others, releasing 5 million barrels of oil into the oceans. The method used to encourage bioremediation of this oil spill involved the use of chemical dispersants at the wellhead in order to facilitate microbial digestion of the oil, in order to keep the oil beneath the surface of the water and away from most of the birds and other organisms that could be damaged by it. These chemical dispersants were designed to take large globules of the spilled petroleum and dissolve it essentially in the ocean so that they formed small globules that could be easily attacked and digested by the bacteria. This did help, but in both cases, the Exxon Valdez oil spill, which is nearing its 30th anniversary, and in the case of the Deepwater Horizon oil spill, beaches and the effects of these oil spills are still being felt. The oil is still in the environment and not completely cleaned up. This despite bioremediation efforts and natural bioremediation. Other microorganisms are used in other ways for bioremediation. This includes Deinococcus radiodurans, pictured here, which is used in highly radioactive nuclear waste plants to digest toluene and toxic mercury. Of course, it doesn't get rid of all radioactive materials, but it can help in bioremediating a few of the chemical components. Bioremediation was also used to remove contaminants discharged as a result of gold mining in an operation in Lead, South Dakota. Now, while microbes have been used to clean up a lot of the pollution that humans have generated, microbes are also useful and have been useful for thousands of years to clean up human natural waste. And we still use them today in our wastewater treatment plants. In our wastewater treatment plants, microbes are used to clean up sewage and turn this sewage water into gray water that can be reused for irrigation and other purposes. There are 3 different stages of wastewater treatment, the 2 of which are almost always used, and the 3rd of which is not always used. The primary stage, or the first stage of wastewater treatment, is when raw sewage is filtered through screens to remove sticks, plastics, and other large debris, and then the wastewater flows into the primary treatment settling tank, where heavy particulate matter, referred to as primary sludge, settles to the bottom. Then, the microbes are put into use in the secondary treatment in order to carry out digestion. Now, this can happen in the process of the liquid portion of the primary tank passing into the secondary tank, which is aerated by a trickling filter. A biofilm composed of bacteria, protozoa, algae, and viruses carry out aerobic and anaerobic fermentation in order to degrade the materials found in this sludge. An alternative trickling system can be activated and used in activated sludge processes. This is done by aerating bubbling air through the secondary tank. Aerobic microbes, particularly in the bacterial species Zooglobia ramigera, grow in aggregates called flocks and degrade the matter. The fluid wastewater from the activated sludge unit passes into the secondary unit and is chlorinated to kill any of the remaining pathogens as a further safety feature. The secondary treatment is successful at removing organic matter from waste, but can become a problem because it does not remove inorganic byproducts of microbial degradation, which may act as nutrients and stimulate algal blooms. Tertiary processes can reduce these inorganic materials in order to discourage algal blooms. However, because this treatment is expensive, it's not always used. Well, that's it for this chapter. And I'd like you to keep in mind, as we go through and discuss how microbes can be deadly and cause disease, that you keep this chapter in mind as all the ways microbes can regularly help us on a day-to-day basis.